Recording pending, it's saying. Why does it say that? I don't want to hit pending. I want it to record now.
Just
there
takes a moment.
We'll discuss and warn everybody. And,
Somebody's running down the stairs.
All right.
You know what? Yeah, Bill just muted himself. I think that's what it was.
We'll talk about muting in just a moment. So welcome everybody to the second webinar of the SUNY Oneonta's OutSMARTing the Pandemic series entitled Vaccines Worth a Shot. I'm Doug Riley, director of the A.J. Reed Science Discovery Center here on campus. I was really pleased last semester when my colleague and friend, Professor Sally Hahn from anthropology, approached me about doing this series. It's been a really, really rewarding project to work on. In the current atmosphere of anti-science, anti-expertise, and anti-vaccination, we both agreed that it would be helpful to highlight the ways in which our own faculty and their expertise can illuminate for us some of the mysteries of the pandemic. In the battle against COVID-19, knowledge truly is power. Just a few housekeeping items as we begin. First, if you are a student and would like lead credit for attending tonight, I'm going to put a link on the chat, which is gigantic, but if you press it, it will automatically register you as having attended. Like I said, it's a big link. If you're not a student and you click on that, you'll get lead credit, but I don't think it does anything for you. Second, we'd ask you to mute yourself during the presentation so we can't hear you running down the stairs. When we have time for questions, you can post them in the chat, or if you want, you can. We'll invite you at that time to unmute, and just like you would at a live in-person event, if you remember what those were like, you can ask it out loud. Finally, I have a few public service announcements. First and foremost, the pandemic is now a year old and it's been hard on everybody. Many people are struggling. For example, over the last year, I've struggled with anxiety in ways that I never had before. Mental health hygiene is a thing, an important thing, and there are resources to help you. In a moment, I'll put a couple links in the chat. The SUNY Oneonta Counseling Center has a very helpful page of resources and information. Let me just grab that link and put that in the chat. And there's always free and confidential help from New York Project Hope. I'll put that in the chat too. The Science Discovery Center has a variety of upcoming virtual events that might be of interest to the campus and the wider community. Tomorrow night, we're hosting a virtual planetarium show at 7 p.m. And on Thursday, March 16th, we're holding an online workshop for our 2041 project. Actually, it's Tuesday, March 16th. Holding an online workshop for our 2041 project, which invites participants to think about a future in terms of climate change that isn't apocalyptic. I'll put a link to all our programs in the chat. All these free gifts I'm giving you. Let's see. Earlier this week, it was announced that New York State will open up a vaccination site in Oneonta on our campus. If, like me, you're eager to know when you can get your shot or shots, be sure to keep up with the most up-to-date information at New York State's COVID vaccination website. We are copying that link into the chat as well. So just to reiterate, it's being hosted on SUNY Oneonta's campus, but SUNY Oneonta is not running it. So, all right, so that's good news. And the other good news is that all of our COVID smart efforts are working to keep COVID numbers down among students and community members. So far, there's been no repeat of fall 2020. It's a cause for celebration of the COVID safe kind. We've come so far and we're not going to let up now. Keep up your smart habits. Socially distance, mask up, answer your daily health screening, routine hand washing and hygiene and testing. We know it's hard, but please stay strong. And we know you try to make the right choices, and we are all very grateful for that. So with that, I'm going to turn it over to my colleague, Professor Han, who will introduce tonight's speakers.
Okay, thanks so much, everyone. So glad to see the attendance here. So I'm just going to make some quick introductions because I really want to just give the floor to our colleagues. Dr. Kelly Gallagher is Associate Professor and Chair of the Department of Chemistry and Biochemistry. Dr. Jill Fielhaber is Assistant Professor of Biology with a research and teaching specialization in microbiology. Together, Dr. Gallagher and Dr. Fielhaber have been sort of a wonder team in terms of science education and outreach. For example, they've been mentoring a team of undergraduate students for the International Genetically Engineered Machine iGEM synthetic biology competition. Apparently they've just come straight here from a talk they just gave about that. And they've also collaboratively led a number of other science education and outreach initiatives for the Science Discovery Center. So we are so fortunate tonight that they are here to share their expertise and inform our understanding of the science of the COVID-19 vaccines. So with no further ado.
Well, thank you very much, Sally and Doug, for inviting us to speak. So I get the pleasure to go first, and just in case my name is not showing up in a way that you can see it, I'm Jill Fielhaber, and so I'm the biologist who will be going first. So the title of our presentation today is called COVID-19 Vaccines Worth a Shot. So I'm going to present the first half of this presentation to you. And as part of that presentation, I'm going to introduce you to a little bit of the sort of basic biology behind vaccines, as well as how those vaccines work in order to help boost our immunity to an infectious agent. And then I'm going to talk about sort of a little bit about the new COVID-19 vaccines, how these vaccines are different from traditional vaccines and sort of what makes them exciting to scientists. And then I will turn it over to Kelly. And so as I'm talking, if questions come up that you have burning in your mind, feel free to put them in the chat. And if they sort of directly relate to what I've just talked about, we'll address some of those at the sort of break between Kelly and my switch. And if they're a little bit more general, and they, we'll hold them until the end, and then we'll answer them then. Also at the end, we'll definitely open it up for some Q&A. So don't feel like you have to get your questions in right now. So I'm going to start a little bit with just some of the basic biology behind how COVID or how vaccines work, period. So before I do that, I just want to talk a little bit about this virus that is causing this COVID-19 disease. Now, in the case of COVID-19, COVID-19 is caused by a virus, something called SARS coronavirus 2, which is abbreviated as SARS-CoV-2. This virus, it causes a disease called severe acute respiratory syndrome, and that's where this SARS comes in. And the virus is part of another family of viruses, which are known as coronaviruses, of which there are several members that have been sort of in the news and in the media over the last 10 to 15 years. So in the early 2000s, there was an outbreak of this brand new virus called SARS virus, which was named SARS after the disease that that virus causes. And so that was our first exposure to coronaviruses. And incidentally, one of the biggest outbreaks of this virus occurred in a city in my home country, in Toronto, Canada. And it was quite big news here in North America. Fortunately, we were able to contain that viral outbreak without sort of too much or too many cases, and that virus went away. Later, a few years later, we had an outbreak which was centered around the Middle East by a virus known as the MERS virus. And it turns out that that virus is also a member of this family of coronaviruses. So really, when it comes to SARS-CoV-2, this represents sort of our third interaction with this family of viruses. So just a little bit about the virus itself. Viruses are super cool, and they're one of my favorite things to talk about in my classes because they are so simple and that they're so good at kicking our butts. Okay, so when you look at a virus at sort of the microscopic, the molecular level, what you find is that there really isn't that much to that virus. Okay, generally, a virus is made up of sort of two things. There's usually a layer to that virus, which is sometimes made of protein, and sometimes it's made of lipid. Okay, and that layer is modified by the addition of all sorts of different proteins. Some of these proteins are really important for the way the virus replicates. And in the case of COVID-19 or the SARS-CoV-2 virus, one of the most important proteins is this protein known as a spike protein. That protein, it sticks out of the surface of the virus and is used by the virus for infecting hosts. Okay, in the case of SARS-CoV-2, there's a bunch of other proteins that are embedded in this outer layer of the virus, which have other functions in the virus's sort of life cycle. And then other than having this layer and some of these strange proteins sticking out of the surface, really the only other component of this SARS-CoV-2 is a genome. So in the case of SARS-CoV-2, here on the inside of the virus, we have a representation of the virus's genome. In the case of SARS-CoV-2, this genome is made up of a substance known as RNA. And it codes for, I believe off the top of my head, something like 15 or so different proteins inside of that genome. So overall, viruses are relatively simple. They're basically a genome, in this case RNA, with a protein layer on the outside of it. And yet somehow these viruses manage to hijack our bodies and make us so sick. Now, the way that SARS-CoV-2 manages to accomplish this is pretty amazing. What happens is when our cells come into contact with SARS-CoV-2, the virus is going to recognize certain cells based on the presence of a protein on the surface of the host cell. So on the surface of a cell in our bodies. Okay. In the case of SARS-CoV-2, SARS virus recognizes this protein. It's a receptor known as the ACE receptor. Now, this receptor is involved in its normal function is regulating our blood pressure. So this receptor is found on all sorts of cells in our body. And so what that means is that SARS-CoV-2 has the potential to infect many different types of cells in our body. Okay. Now, this explains sort of why SARS-CoV-2 or coronavirus symptoms are so variable. Some people get just respiratory symptoms. Other people have gastrointestinal symptoms when infected by the virus. Some people have pretty severe changes in blood pressure when infected with the virus. And in part, that's due to the types of cells that the virus is infecting. So these receptors are important because the receptor is involved in helping or is used by the SARS-CoV-2 virus in order to infect a host cell. So these spike proteins shown in blue on the surface of the virus are going to come into contact with this receptor and essentially trick our cells into allowing the virus to enter the cell. Once inside of our cell, that virus is going to hijack the host cell. and use the host cell's machinery in order to make new viruses. Okay, so what will happen is that the virus will enter the host cell, and then this protein coat on the outside of the virus disintegrates. And what that does is it releases the virus's genome into the inside of the host cell. Inside of that host cell, that virus is going to replicate itself. Okay, and essentially genes in this viral genome are going to cause the host cell to begin to make viral components based on the sequence of genes inside of this genome. So some of those genes are going to guide the cell to make copies of this viral genome, so this viral RNA. And so you'll wind up with many copies of the virus's genome. That's one part of the virus. Okay. Other genes in this viral genome are going to cause the cell to use the gene sequences in this viral genome in order to build the proteins that make up the virus. And so what you wind up with is you wind up with a cell that becomes a little virus factory, building the two main components of a virus, the virus's genome and the virus's protein. Once there's enough of these materials created by the cell, well, then those components are going to begin to assemble and form new viruses. And so this causes this host cell to begin secreting and making new copies of this virus. And so that leads to a spread of the viral infection inside of our body. So that's how SARS-CoV-2 works at a cellular level. What does this mean for our bodies as a whole? Well, what happens is that first off, we have to come into contact with the virus. And the most common, based on what we understand about SARS-CoV-2 right now, usually contact comes in the form of inhalation of viral particles, which is why it's so important that we wear masks in order to help block the transmission of this virus. But every once in a while, an individual will take a big breath of air and will inhale a lungful of virus. And what will happen is that that virus will begin to infect those cells in the lung and begin to produce more viruses. So that's what's happening on the virus's end. What's happening on the immune system's end? Well, our immune system is going to begin to fight the virus. And so as that virus is replicating inside of our lung tissue, and this is illustrated here, this is an alveolus, which is one of the pockets in the lung where gas exchange occurs. It's basically a hollow sac lined with cells that's surrounded by blood vessels. Okay, so that gases and materials can move in and out of the bloodstream to be inhaled or exhaled. So while that virus is replicating and copying itself, it's doing so inside of the cells that line the alveolus, and new viral particles are being produced. Our immune system is going to begin to fight the virus. And so what will happen from our body standpoint is that white blood cells from our circulation are going to move from the blood vessel into the airway space inside of the alveolus. And those white blood cells are going to begin to actively fight the virus. Okay, they'll recognize the virus and then begin to produce substances which will help support the recruitment of other immune cells. They're going to support inflammation, which is going to increase blood flow to the site of the infection, so increase blood flow to the lung tissue. And also some of those substances that those immune cells are going to produce are going to increase the permeability of the barrier between the alveolus and the blood vessels. And so that's going to make it easier for blood cells to enter into the airway space. Now, this immune response is responsible for some of the symptoms we have of COVID-19. So things like fever. That's part of our normal immune response. That sort of wet hacking cough that many COVID-19 sufferers experience is due in part to that increased permeability. Okay. In mild cases of COVID-19, the immune system fights the infection and then resolves the infection, and those symptoms go away. But in severe cases, those symptoms have the possibility of leaving or affecting other organ systems. And so what sometimes happens in severe cases is that some of these inflammatory substances that white blood cells produce, which are called cytokines, those substances can enter the bloodstream. And when they do, they have the ability to come into contact with other organ systems. So for example, they can interact with liver or kidney cells. And this can lead to inflammation and symptoms such as liver damage or kidney failure in severe cases. Okay. That being said, that immune response is really important. That's what we call the innate immune response. That immune response is absolutely critical for the sort of next phase of the immune response, which is known as the adaptive immune response. In the adaptive immune response, we activate specialized cells, which act as they are really good at removing infectious agents from our bodies. Okay, so what are some of the aspects of this adaptive immune response? One aspect of this adaptive immune response is something called humoral immunity. Humoral immunity is a type of immunity which is controlled by a specialized type of adaptive immune cell called a plasma cell. What plasma cells do is these cells, they create antibodies, which probably everybody has heard of antibodies at some point, but just in case you haven't, these are Y-shaped proteins that these plasma cells will produce when presented an infectious agent by the innate immune response. When these antibodies are produced, what they do is they bind to any virus particles that they encounter and they neutralize them. So basically, any free virus that has left a cell after being made has the potential of encountering these antibodies, which are going to bind to the virus and essentially prevent that virus from infecting a new cell and spreading the infection. And once bound by these antibodies, that inactivated virus can then be consumed by other white blood cells and removed from the body. So that's one arm of this adaptive immune response, and it's really effective when the virus is outside of a host cell. Okay? But virus is sometimes found inside of a host cell. So that means that humoral immunity is not sufficient for getting rid of a viral infection. We also need another type of adaptive immune response, something called cell-mediated immunity. Cell-mediated immunity is effective against virus when it's inside of cells. So while the virus has infected a cell and is causing that cell to make more viruses. Okay. This type of immunity is mediated by another type of cell called a cytotoxic cell or a killer cell. Okay, these killer cells, their job is to recognize and kill any virus-infected cell it encounters. So that way, we have cells that are fighting the virus while it's inside of the cell, and we're fighting the virus while it's outside of the cell. So these two branches are incredibly important. As part of cell-mediated immunity, we also get the activation of a third type of adaptive immune cells, something called helper cells. Helper cells secrete some of those cytokines that were present in the innate immune response. These cytokines support the actions of these cytotoxic cells and these plasma cells, helping the cytotoxic cells be more effective killers and helping the plasma cells be more effective antibody makers. Okay. Now, a key feature of this adaptive immune response is that both humoral and cell-mediated immunity also produce a specialized type of immune cells known as memory cells. Memory cells are incredibly long-lived types of cells. Okay, most of these types of cells, these helper cells, these cytotoxic cells, and these plasma cells, they only live a few weeks in our body, and then they die and new cells emerge. But these memory cells can live for much longer than a few weeks, anywhere from about 1 to 10 years, depending on the memory cell. Okay. This is really important because this is a way for our immune system to remember any invading or any virus or any invading pathogen that we might have encountered. Now, when we respond to something like a virus the first time, we produce these memory cells. These memory cells, their job is to live in our body for a long time and respond more rapidly the next time that virus is encountered. Okay, so these guys respond more quickly than the first time. The first time an adaptive immune response is activated, so they respond more quickly the second time. And they also have a stronger response. So they produce better antibodies and they produce better cytotoxic cells for killing infected cells. Okay. So how do vaccines work? They work by stimulating our immune response. So a vaccine mimics the process of a natural infection. So when we get a vaccine, that vaccine contains substances which resemble whatever the pathogen is or whatever the organism that causes disease is, in order to elicit an immune response without that infectious agent. Okay? The goal of vaccination is to elicit an immune response. The reason is to build these memory cells. So that way, we have a population of these long-lived cells with a stronger response that are going to respond to that agent if we ever encounter it again. Okay, so that's the basic principle of vaccination. Okay. Now, here we have a couple of graphs which sort of illustrate the course of an infection. So let's look at the graph on the left-hand side of the screen here. So what we're doing is we're sort of graphing the magnitude or how strongly the immune system responds, okay, to an infectious agent over time. Okay, so what we find is that for a generic infection, and this is true for basically any infectious agent, okay, when we come into contact with that infectious agent, within anywhere from 24 to 48 hours, we begin our first initial responses, our innate immune responses. The function of that innate immune response is to just kickstart our immune response so that it tells our bodies to begin to activate that adaptive immune response. This response tends to be sort of high magnitude, but is very limited in time. Okay, so high magnitude, for example, during this time, you might have a fever, and it might be a fairly strong fever. That's what's going on right here. We have this strong innate immune response. And the function of that is to help limit or help slow down the spread of that infectious agent. Okay. As that innate immune response sort of ramps down, the adaptive immune response begins to ramp up. And so what we see is after that sort of initial burst of symptoms and magnitude, slowly the adaptive immune response begins to respond. And what we'll see is that antibody levels in the body begin to rise, and eventually they reach sort of a stable level. Okay. Likewise, the same is true for the production of those cytotoxic cells and those helper cells, which are shown in the blue line here. They sort of go and they sort of ramp up and then they sort of level off. And so it's this adaptive immune response that is actually going to remove that invader from our bodies. And then over time, once that microorganism is destroyed from our bodies, then those responses will decrease. The same thing is true for the average SARS-CoV-2 response. So initially, a virus will begin to replicate inside of the body, and the numbers of viruses, which we call the viral load, will begin to go up. And there'll be a little bit of a lag, but the innate immune response will kick in and will begin to fight the infection. In the case of SARS-CoV-2, that innate immune response is sometimes not as strong as might be present in sort of your generic infection, because the virus has mechanisms for sort of interfering with that response. But for the average infection, you definitely get a response. So that innate immune response will kick in, but the viral load will continue to go up. It's not until we get that initial kick of antibodies and cytotoxic and helper cells, as that begins to go up, what we see is that the viral load will sort of reach its apex at its highest level. And then eventually the sort of battle tips and our immune system will begin to win out and the viral load will decrease. So this is for your sort of average SARS infection. In the case of severe SARS infections, what you often find is that those severe infections are characterized by a very weak innate immune response. So instead of reaching sort of this magnitude, the sort of peak of the innate immune response might be down here where my pointer is pointing, so fairly low level. Now that low level innate infection response is not good because it allows the virus the viral load to increase more rapidly, and it delays the adaptive immune response. And so the goal of vaccination in the context of SARS-CoV-2, particularly when it comes to the severe SARS-CoV-2, is that by using a vaccine, what we're doing is we're shifting the immune responses, okay, over in this direction. So the idea being is that we're going to pre-elicit an innate immune response, and that pre-elicited innate immune response is going to trigger the adaptive immune response long before we come into contact with the virus. And that means that as soon as that virus comes into contact with our body, it's already encountering a body that is primed to actively seek and destroy the virus. Okay. So, okay. Little bit now, we've got some basic biology behind us, and so now we can look at some of these new COVID vaccines and how do they work and what's new about them. So before we do that, we should talk a little bit about some different vaccine platforms. So what I mean about vaccine platforms is I mean how are vaccines that we're already familiar with actually manufactured. Okay, so historically, sort of up until the last 5-10 years, there's really been 4 main ways that vaccines are manufactured. One way is through the use of a whole inactivated virus. So to make this type of vaccine, what you would do is you'd grow up virus in chicken eggs, And then you'd isolate the virus from that egg product, and you'd inactivate the virus by, exposing it to chemicals that that destroy parts of the virus. And so that makes the virus inactivated, so it's not able to establish an infection. But that being said, the virus still the inactivated virus still resembles the original virus. And so when you inject or you administer that inactivated virus, it resembles the living virus enough so that it can trigger an immune response. These whole inactivated vaccines have been around for many, many years. An example of one such vaccine, which if you're one of our older audience members, you may have actually had in your lifetime, is the polio vaccine. So that's an example of a whole inactivated virus vaccine. Related to that is another type of vaccine, which is made by using something called a whole attenuated virus. A whole attenuated virus, the word attenuated means weakened. Okay, so in the case of an attenuated virus, what you might do is you might take a virus that infects humans, and you might force it to grow in a mouse for many, many viral generations, for years and years and years. And as that virus is growing in that mouse, over time, that virus weakens and it loses its ability to effectively cause infections in humans. So then you can take that weakened, attenuated virus and you can use that as a vaccine. And again, because the virus resembles the original virus, it can elicit immune response to the original virus. So an example of a vaccine that's made this way is the measles vaccine. The measles virus has been attenuated through many years of growth in another organism in order to weaken it. Another type of traditional vaccine platform is a protein subunit vaccine. So in a protein subunit vaccine, you don't take a whole virus, or a whole bacteria to make the vaccine. You take one little protein from that organism, and you grow up samples of that protein in the laboratory, and then you use that as your vaccine. And so the immune system recognizes that one protein. So an example of a vaccine made this way is the tetanus shot, which is made against tetanus toxin, which is a protein produced by some bacteria. And then finally, sort of the last traditional vaccine platform that is frequently used is the generation of virus-like particles. And so in a virus-like particle, what you do is you make a vaccine which represents that outer layer of the virus, but you make the virus particle empty. So there's no copy of the virus's genome inside of that particle. And so that's a virus-like particle, but it's not a virus. So that way you can inject this into the body will recognize the outer coat of the virus, but there's no viral genome inside to result in the hijacking of a host cell. And an example of a vaccine made this way is the human papillomavirus vaccine, which many of our younger audience members would be familiar with. So these are sort of the traditional vaccine platforms. We have new vaccine platforms as well, which have really sort of come into their own or are coming into their own right now. One example of such a new platform is something called a viral vector vaccine. Now, a viral vector vaccine, to make this type of vaccine, what you need to do is you need to take a relatively harmless virus. So something like a virus called adenovirus, which is one of the viruses that causes the common cold. It's a virus, but it's not one of the more serious ones that we would think of. And what we do is we take the outer coat of the virus, that harmless virus, we remove parts of that harmless virus's genome, and then we put in pieces of the genome of the virus that we want the vaccine to be effective against. So, for example, one virus vaccine that's made this way is something called the Ebola vaccine. In this vaccine, you take this adenovirus and you'd essentially add a couple of Ebola virus genes to the genome of this virus so that this harmless adenovirus or this weak adenovirus can induce an immune response to those Ebola genes. So now you can inject this virus and the body will mount an immune response against these Ebola genes. But because it's only a few genes and it's not the entire virus itself, you have no risk of catching Ebola from this vaccine. And you can actually mount an immune response to that virus. We have other types of vaccines that are in development but are not yet licensed for use in humans. One such type is a type of vaccine called a DNA vaccine. A DNA vaccine is basically a piece of DNA, which is the stuff that our human genome is made out of. And there's a carrier component, which is shown in sort of the lighter gray. And then you take the gene, a couple of genes from the virus that you want to elicit immune response against, and you put them in that DNA, and then you can use that DNA as a vaccine. While in development, there are currently no DNA vaccines which are yet licensed for use, but this is an emerging technology. Likewise, there's another emerging technology which involves taking some of the immune cells of the innate immune response, a type of cells known as antigen-presenting cells. These cells are involved in turning on the adaptive immune response. So in this type of vaccine, you take some antigen-presenting cells, and in the laboratory, you teach these antigen-presenting cells to recognize and display parts of the virus on their surface, so that you can then take these antigen-presenting cells, inject them into a human, and those antigen-presenting cells will show these pieces of the virus to the adaptive immune response. And that will help trigger that adaptive immune response. Again, these are not currently licensed, but this type of technology is licensed for use in cancer therapy. So this is definitely an emerging technology. And then finally, we have our RNA vaccines. And so these are the types of vaccines that the SARS-CoV-2, some of the SARS-CoV-2 vaccines are. This is a very new technology, and it's very exciting. This is the first, the SARS-CoV vaccines that use this technology. This is the first time this technology has been licensed for use. And so I'm super stoked about this because I've been watching this technology develop basically my entire career. In these types of vaccines, what you do is you take a piece of RNA, okay, and into that RNA you add some of the genes from the viral genome. So in this case, some of the genes from SARS-CoV-2. Okay, and specifically the spike protein gene. So the gene that codes for that spike, which is on the outer surface of the cell, which is used to recognize that ACE2 receptor on the surface of the cell. This piece of RNA is then packaged up in a lipid particle and then can be delivered through an injection. And so the body will recognize this RNA and use that RNA in order to make some of these viral proteins and elicit an immune response against those viral proteins. So that allows the body to pre-recognize that viral spike protein. So As for the vaccines that are currently approved for use in the United States, we have two different types of vaccine platforms which have been approved. Okay, the first, or one, I guess we'll say not the first, but the most recently approved example of a viral vaccine, COVID-19 vaccine, is the Johnson & Johnson vaccine, which was just approved, I think a week or two ago. Time sort of bleeds together at this point. Okay, so in this type of vaccine, this J&J vaccine is a replication deficient viral vector vaccine. So if you'll recall, this means that we're going to take a harmless virus and we're going to engineer that virus so that it produces some of the proteins from the SARS-CoV-2. So that's how the Johnson & Johnson vaccine works. So you build that viral vector vaccine, which contains the coat of the harmless virus, but then some of the coronavirus spike DNA, and you inject that into a host. Now that replication deficient viral vector, while that virus can't copy itself, what it can do is deliver that genome, that DNA, or pardon me, yeah, that genome into the host cell. And so the harmless virus acts as the delivery mechanism for the genes that you want to elicit immune response against. Now, those genes are in DNA form, okay? And so for the cell to use those genes in order to elicit an immune response, we need to express those genes. And so that's going to involve the host cell making a substance out of, or basically reading the sequence of this gene right here, which is coded for in the DNA, and using it to create an intermediate molecule known as a messenger RNA, or mRNA for short. That messenger RNA can then be used by a protein-making factory known as a ribosome. The ribosome will read the sequence of that messenger RNA and use that to create the coronavirus spike protein. Now, remember, the coronavirus spike protein is just the outer layer of the virus. And while it's important for the virus to replicate, it is not a complete virus, and so it's harmless. But now we have cells making this protein inside of our body, and this protein can elicit that immune response that we're looking for. So that's how the J&J vaccine works. That technology has been around for a while, and so it's super cool, but it's not quite as exciting as the other two vaccines, which are based on messenger RNA vaccines. Now, in these messenger RNA vaccines, and this is the Moderna and the Pfizer vaccine, these mRNA vaccines, if we just go back one slide, an RNA vaccine is made up of a piece of RNA surrounded by and a delivery lipid, and that's exactly what we see here. So the mRNA that is inside of this delivery lipid codes for the viral spike protein. So in this vaccine, you inject this vaccine into the body, and what will happen is that this lipid nanoparticle, which surrounds the messenger RNA, is going to fuse with the lipids of our cells. This technology has been used in the laboratory for delivering nucleic acids to cells for many, many years now, and so we know that this will happen. When that lipid particle fuses with our host, our cell membranes, that delivers that messenger RNA to the cell. Now, this messenger RNA is used the same way a messenger RNA is used over here. So the ribosome can read the sequence of this messenger RNA, which was delivered by the vaccine, and it will produce coronavirus spike proteins, which will elicit an immune response. The advantage of using this technology is that messenger RNAs are naturally short-lived molecules inside of our cells. So our cells make messenger RNAs all the time, but they tend to not survive inside of the cell longer than, 24 or so hours. And so you get this short burst of making these coronavirus proteins, and then this messenger RNA degrades. This is advantageous because the messenger RNA, it does not resemble our genome. So there's no risk of accidentally recombining a piece of DNA with our genome. And so that's a really attractive feature of these vaccines. So I've been talking for quite a while here, and I've come to the end of my section. So I am going to stop and I'm going to let Kelly talk.
All right. I think we were going to, oops.
Oh.
Let's see, stop sharing.
Did I? Oh, I didn't. Sorry. I'm going to stop sharing right now.
All right. I think we were going to pause and see if there were any questions that had come up to this point.
Nothing in the chat.
Okay.
All right. Then I'm going to share my screen. Let's see. All right, so Jill has told you about the basic biology of how these vaccines work and or the theory behind how they would work. So what I'm going to tell you is the kind of practical side of things, how we actually took this science and turned it into a product that is going into people's bodies right now and helping us recover from this pandemic. So I'm going to talk about how the vaccines were developed as a product and how they were tested to ensure that they were safe and effective. So what we have here is the timeline for how a traditional type of vaccine would be developed. So there's lots of different stages going from, beginning from, let me see if I can get a pointer here. Beginning from, non-clinical research here, okay, which means we're studying the basic biology of the virus. We're learning what the molecules of the virus look like. We're studying how it infects cells, what its targets are, and understanding sort of the enemy that we're trying to defeat. Okay, and this process can take, 3 to 8 years on this timeline. So at this point, we're trying to learn about the virus and then come up with a philosophy for how we're going to design a vaccine. And then we're going to start what we would call the clinical trial phase or the clinical research part of developing a vaccine. So the clinical phase takes, up to 10 years in the past. It has taken up to this length of time. And it has 3 parts to it. And they're known as phase 1, phase 2, and phase 3. So in phase 1, we do what are called human pharmacology studies. In this case, up to this point, we have our vaccine. It's been tested in animal models. It's been tested in cells in what we call cell culture. So not in an animal, we can grow them in petri dishes. And so we know it behaves a certain way outside of humans. Now, in phase 1, what we're going to do is begin the process of testing a vaccine in humans. The first thing we need to do is to just make sure that the thing isn't a poison. So human pharmacology studies that are done in phase 1 are not at all concerned with, does the vaccine prevent infection? What they're concerned with is, when we inject this into humans, does it make them sick, or is it toxic in some way? So after we have done the human pharmacology studies, then we get to proceed to phase 2. In phase 2, we start to investigate whether the vaccine has any therapeutic benefit. We're not looking to determine exactly how effective it is. We're looking for signs that it will be effective, and we're playing around with the parameters of the vaccine. We might be adjusting dosages, or looking at, do we need 1 shot or 2 shots, and how far apart should they be? And so there's a combination, again, of having, putting the vaccine into volunteers and then examining the effect of the vaccine on their immune response. After phase 2, then we can proceed to phase 3. And phase 3 is the phase of vaccine clinical trials, where we ask the question, is this vaccine effective? Is it going to prevent people from getting sick? And we do, again, additional safety studies looking at side effects. So at this point, we take a particular dosing regimen that we identified in phase 2, and we apply it to a large group of people who had volunteered for this clinical trial. And I'll talk a little bit more about that in a few minutes. So once we have made it through all 3 phases, there's a lot of data that has been generated. And typically a vaccine is not authorized until scientists and specialists at the Food and Drug Administration have had time to evaluate that data. Okay, so that can take between 1 to 2 years. At the end of that process, okay, a vaccine would be approved. Now, most of the time, companies have invested millions of dollars up to this point on these phases. But in the past, companies would not invest in mass production of the vaccine until they got approval. Okay, so this is going to be something that we find is different with the COVID vaccines. So after approval, so this would be the approval point, the vaccines are going to be manufactured, and then they're going to be available and put into people's arms. Now, we continue to study vaccines after they're authorized, just to get more and more data, and so that we have a better understanding. Okay, so this line right here, when the vaccine is available for use, at this point, it has taken anywhere from 6 years to 20 years for a vaccine to be developed. Now, with the case of coronavirus or COVID-19 disease, we did not have the luxury of having this timeline. And so a variety of things were done to accelerate this timeline. And because of that, we were able to vastly speed up this process. Okay? So watch very carefully as I transition the slide, and you'll get a sense of how the process was shifted. All right, so you'll notice that all those colored bars shifted left. So there are a couple of things that enabled us to go from what could have been a 20-year process to 10 months and 15 days before a COVID vaccine was given emergency use authorization. So what were the things that contributed to this? Well, before we talk about the development pipeline and how that contributed, I have to point out that this phase here, okay, the science phase, was vastly accelerated by the fact that a decade ago, there were outbreaks of the SARS and MERS viruses that Jill had mentioned before. So we had seen a coronavirus disease, and it was severe but short-lived. And there have been, smaller outbreaks from time to time of these diseases, enough to cause concern for people to study them. Okay, and so there is, in fact, and has been a decade's worth of research, a decade plus of research on coronaviruses. So the virus that's currently causing the pandemic is significantly different in a few ways, but it has enough similarities, okay, that the process of learning about the virus that was causing this disease was vastly accelerated. So when you're a scientist who's studying a virus, one of the things you have to figure out is how you're going to do your experiment. And that's a process that could take years. How do I grow this virus? How do I, what animal model can I use? Because we had studies with other coronaviruses, we knew sort of the basics of how to handle this virus. So a lot of the experiments that we were able to do were just taking the methods that have been developed over 10 years and applying them to this new thing we were seeing, right? And the next thing about the basic science that I want to say is that we really owe a great debt to scientists like Kizzmekia Corbett, who is at the NIH, and she was involved in the development of the Moderna vaccine. But Kizzmekia was a scientist who went to the NIH right after she had finished her graduate work to do something called a postdoc. And when she got there, she told her mentor that she wanted to study coronaviruses, which were not really popular at the time, but she was kind of savvy. She was interested in the biology of coronaviruses, but she also knew that nobody else is studying these, and these have caused some minor outbreaks in the past, and they might have the potential to in the future. So she worked for many years just based on that curiosity and trying to understand how this particular type of virus worked. And so she and her lab, the scientists who worked with her, were able to step up when the world had this great challenge and do a lot of science in a very short period of time. So the basic science was primed to help us understand what task needed to be done. In another strain that, there's been 15 years of development of these mRNA vaccine concepts that Jill was talking about. The other key thing that accelerated our timeline, again, from, decades to less than a year, is the fact that government agencies and pharmaceutical companies and academic labs all agreed to take a new approach to developing a vaccine. So you'll notice, if I go back to the previous slide, you'll notice my phase 1, my phase 2, and my phase 3 were all successive. Each one followed in a series. Phase 2 did not start until phase 1 was done. You'll notice that the COVID vaccines, okay, a lot of these bars are overlapping in time, okay? So the approach that was taken with the COVID vaccines was to do some things that we traditionally do one after the other at the same time, right? So while we were doing, our pharmacology studies and our exploratory studies, we were also doing smaller clinical efficacy studies that then we were able to ramp up into much bigger efficacy studies, once we had finished these 2 phases. The other thing that happened is that government agencies agreed, because of the emergency status, to continuously monitor the data. So what was happening was this loop of clinical studies, initial clinical studies being done. The data was continually being sent to the FDA, who was providing feedback to the companies developing these vaccines. And then the final thing that really accelerated the availability of these vaccines is, you'll notice that this production part here, which never overlapped with any of the other parts, now overlaps with the phase 3 trials. Okay, so once Pfizer knew that the vaccine had passed phase 1 and it was not going to be toxic, all right, they knew that there was going to be a huge demand for the vaccine if it was effective. And they had enough data from their animal models and their early clinical trials to be willing to bet that the vaccine was going to be effective enough to be useful. And so large-scale production methods were developed while clinical trials were still going on and actual production was starting. So once we hit this point where we got emergency use authorization, they had a stockpile of vaccine. So, all right, so we've now developed this method that in the case of an emergency, we can get a vaccine in a very short period of time. But the question then becomes is, well, how do we know these things work? And when I say a vaccine is effective, what does that mean? So you've heard, probably everyone's heard, that the Moderna and Pfizer vaccines are 95% effective, 94% effective. The J&J vaccine is 66% effective. What does that mean when we give those percentages? Does it mean that, in the case of Pfizer, which is 95% effective, does it mean that 5% of the people who get the vaccine are still going to be able to get coronavirus? Does it mean that anybody who gets the vaccine could still get coronavirus, but it will be 95% less severe? There's lots of different ways you could interpret that number. So I bet you didn't think you were coming to math class, but I'm going to use a little case study example to explain to you what it means when we say a vaccine is 95% effective. So we determine vaccine efficacy in phase 3 clinical trials. And what we do is we get a large group of volunteers who are willing to participate in this study. So the numbers on this slide are from an actual Pfizer vaccine study, right? And so they had 36,000 participants and they divided them into 2. All right, and this group got the vaccine, and this group over here, which had 1 fewer person in it, got what's known as a placebo. So they were given an injection of something, but it contained no active ingredient. Okay. Now, when we do these types of trials, they're what we call randomized and double blind. So there's no strategy as to who goes in which group. The individuals in the trial do not know whether they got the vaccine or the placebo, although in this case, many of the volunteers were told that at the end of the study, they would be told if they got the placebo and offered the vaccine, which is a little different than how things are normally done. And then also, in addition, the physicians and healthcare workers administering the vaccine did not know if they were giving someone the placebo or the vaccine. Okay, So everything is coded so that only after the data is collected can we deconvolute it and know, who had what. So they followed these 36,000 people for 120 days, asked them to live their lives as they had been, wear your mask, do your social distancing. And what they did is they counted the number of cases of coronavirus that occurred in each group. Now, these were people that were in a trial that were being closely monitored for 120 days. So they were regularly tested for the virus so that the people doing the study would know, without a person having to get sick, whether or not the person was infected with the virus. So what we call the primary endpoint of this trial is the virus able to infect and copy itself in this person? It had nothing to do with whether or not they got sick. So basically, without going through all the details of math, you have a rate of infection for each group. How frequently did a person get infected in each of the two groups? You compare them, and then you turn that comparison into a. So what this tells us is that compared to people who were unvaccinated, okay, there was a 95% lower chance of getting infected if you had the vaccine. Now, the interesting thing with these vaccines, in terms of a secondary endpoint, which is whether a person was feeling symptoms or not, we found that these new mRNA vaccines were 100% preventing people from getting severe disease, which was not the case in the placebo group. Okay, so it's good news on two fronts. One, most of the people are not going to be infected at all if they're vaccinated. And two, if they're vaccinated, even if you were to somehow get infected, you're not going to get severe disease. And so that's a great outcome. So to put things in the context of vaccines that you may be familiar with, and ones that you have been administered in your lifetime, okay, we have the new COVID vaccines, which are up on the top, and then things like the flu vaccine, chickenpox, measles, the oral polio vaccine, and tetanus. So what you'll see is that in terms of efficacy, okay, the new vaccines compare very favorably with the old style vaccines. Okay, certainly the mRNA vaccines have very, very high efficacy rates. And the Johnson & Johnson viral vector vaccine is 66% effective. And that may seem like it's a lot worse than the Pfizer and Moderna. But there are a couple of advantages that the J&J vaccine has that this chart is also designed to illustrate. Okay, one thing you'll notice is that I've color-coded this chart. Okay, so a practical aspect of developing a vaccine is how you're going to store it and how you're going to get it to people. So what you'll notice is that everything that's in white gets stored at 2 to 8 degrees Celsius, which a refrigerator is 4 degrees Celsius or 40 degrees, about 40 degrees Fahrenheit. Okay. Now, there are some vaccines of, even the traditional type that need to be frozen. So minus 20 degrees, okay, is your freezer. Okay, so your typical kitchen freezer is minus 20 degrees Celsius, which is, about minus 4 degrees Fahrenheit. Okay. Again, still not bad because you can, everybody can keep it in their kitchen. Now, the Pfizer vaccine, which is again one of these mRNA vaccines, needs to be stored at minus 70 degrees Celsius, which requires a special research grade freezer. So we have a disadvantage, okay, in terms of if you're trying to get this vaccine to the whole world, okay, you're going to need something that can be administered in developing countries. So if you look at the Johnson & Johnson vaccine, okay, in terms of what the FDA thinks is an effective vaccine, anything over 50% would be acceptable and considered good for the control of coronavirus disease. I see that Doug has asked how effective Johnson & Johnson is at serious disease. And I'm going to get to that in a second. So you'll notice that Johnson & Johnson only has one shot. It can be stored in a refrigerator. And of the new vaccines, it's the lowest cost. Okay. So Johnson & Johnson did its phase 1 and phase 2 studies. And it tested whether or not to do a 2-shot regimen. When they did a 2-shot regimen, okay, they found they had 100% efficacy in terms of preventing disease. However, and I'm guessing at this, but my guess is that they realized that the world needed a solution that would be usable in third world countries or developing nations, where having people be able to access a vaccine on multiple days, a month apart, and storing things at super cold temperatures would be very difficult. So they found that their vaccine was successful enough. It was 66% effective at preventing any disease. And just like the Pfizer and Moderna, it's about 100%, 95% effective at preventing severe disease. So we have something that's going to do the job. And there are some trade-offs in terms of preventing any disease, but it's something that can be deployed in areas that are not as prosperous as the United States or not as technologically advantaged. So really, there's a lot of things to consider when you're trying to develop a vaccine. And, sometimes you don't have the luxury of saying, I want 100% efficacy, because there are other things to consider, whether or not it can be manufactured in a developing country. So the other thing that comes up and has been in the news a lot lately, and I'm going to go over this just briefly, is you may have heard of variants coming up. And there's, some people have expressed concern that the vaccines we have are not going to be or might not be effective against those variants. Okay. So this chart just shows you just a few. There are hundreds and hundreds of what we call coronavirus variants. It's basically, if you were to determine the genetic sequence of the virus, the entire virus, would it be different than the first one that was isolated from Wuhan, China? Now you'll notice on this chart that the names of different geographic locations are in parentheses. That's because you will hear in the news them talking about the Brazil variant, the UK variant, the South African variant, the original virus isolated in China. And in the virology community, we try not to associate viral strains with places. And it's not just out of a concern for not casting aspersions on a particular portion of the world. It's because of the very real scientific fact that just because we found this B117 variant in the United Kingdom first, it doesn't mean that the first person to ever have that variant wasn't from the United States, or it could have been from Italy. It's just that it was first observed in that region. So we have, for all these variants, and there are hundreds, there are numbers, but some of them are more significant than others because the virus structure changes just enough for it to behave a little differently. So, for example, these particular variants are being investigated for whether or not the virus gets into cells better, or whether an antibody that's directed at that spike protein is not as effective at preventing infection. So there's both, good news and bad news in that the current viruses that we have are effective against some of these variants, but there seems to be some reduced efficacy. Now, one of the reasons that it's so important for us to still wear our masks and still socially distance is because these variants arise because this virus has been allowed to run amok in the human population, because we didn't control things very early on. There were lots of people infected, and every time a virus infects someone and makes copies of itself, there can be changes when it makes those copies. So it's not like a Xerox machine that automatically makes an exact copy. Sometimes there are little mistakes and changes that happen, and sometimes those changes result in a change in something like the spike protein being slightly changed in shape that might make it more virulent. So the sort of last thing I wanted to talk about was why scientists and virologists and people in healthcare are so excited about mRNA vaccine technology. And the reason is because mRNA vaccines can be faster, cheaper, and more potent than traditional vaccines. So again, we're going to go back into the process of how these things are manufactured in this table to just explain why this is the case, okay? So traditional vaccines, okay, so something that uses a part of a virus that you have to grow up, okay, again, development takes 10 to 15 years. The mRNA vaccines, again, they can be developed very fast. And one of the reasons that you'll hear people talking about, well, if the vaccines aren't as effective at at protecting against the new variants, well, we might have a booster. Well, it's very easy to produce a booster of an mRNA vaccine. If you remember, it's just a piece of nucleic acid, RNA, that can be chemically synthesized, all right, in the lab, just encased in a lipid coat, which is just basically fat, okay, a special kind of fat. So very easy and very quick to just change the chemical synthesis. Let's say there's a change in the spike protein at this point in the sequence. We just, the next time we synthesize a dose of that vaccine, we just change the nucleic acid letters that's there, and now we have a vaccine against that variant. So we can respond very quickly. All right. Now, because this is more of a chemical type of process of producing these vaccines, we can standardize it, okay? And the only thing that's going to change from, MERS to SARS-1 to SARS-2 is going to be, what are the letters on this RNA strand? Whereas for the traditional vaccines, you have to customize your process development timeline so that you may not be able to grow your virus in eggs, like Jill said. You may have to grow it in cells that are in culture, and that takes a lot more time and effort. Because your process is going to be customized for each vaccine, what happens is, if you want to expand capacity, then you have to build, a new factory for each type of vaccine you want to make. Whereas in the mRNA vaccine world, wing 1 can be for SARS, wing 2 can be for MERS, and wing 3 is we need more SARS, right? So it's the same type of equipment, the same type of facility. You just literally change a code in your computer program. manufacturing costs orders of magnitude lower. And then the other thing in terms of the practical aspects is because this is such a simple thing, okay, you can, we can stockpile the active ingredient, and the active ingredient in this case is RNA, in teeny little vials, and you can have it frozen, and then you can have thousands and thousands of doses in very small volume, and you can just formulate them at that fatty coating before you're going to send them out to the customers. Whereas, again, traditional vaccines physically take up more space and are going to, need more resources to store. And then the final advantage is getting back to the science, is that traditional vaccines only do that humoral response, not the cell-mediated immunity. So that original slide Jill had with the mouse, only the left-hand side of the immune system is activated, not the right-hand side of that slide. Whereas the mRNA vaccines develop both of them. And then the other thing is there's something called an adjuvant that is required. So you can inject something into someone's arm or muscle, but is your immune system going to react to it? So there are chemicals that are typically added to vaccines that are used to kind of irritate the immune system so it reacts. And these are often the chemicals that people who are concerned with vaccines point to as one of their reasons for concern. Now, we've used them for over 100 years, and they're generally safe. But if you don't have to use them and you get an immune response, hey, that's an advantage both in production cost and, potential safety of the vaccine. And so mRNA vaccines have been shown that they don't need those chemicals. Okay, there are features of viral RNA that on their own kind of help activate your immune system. So again, mRNA is, faster, cheaper, and again, they do more for our immune system than traditional vaccines. So that's one of the real reasons why we're so very excited about the fact that this technology is being used and it's working. So anyway, so that was all we had to present. But before we take any questions for anybody who's stuck with us this long, I wanted to point out that today was a very fortuitous day for us to do this talk, because the spike protein, which are those things that stick out from the virus, is the target of all of these vaccines. And exactly 1 year ago today, the scientists published for the very first time what the shape of that molecule was. So I've made a representation here, and this is shown in a way that biochemists typically represent protein structures. So exactly 1 year ago today, we found out what our target looked like. So pretty exciting. All right, and I'm going to stop sharing. And all right, so if anybody has any questions.
We got a good question here from Tracy Allen.
Yep.
So
so I don't know that the question is, why is the Johnson & Johnson adenoviral vector less effective than the mRNA vaccines? And so clearly no one understands exactly why at the moment, right? There's a great podcast that I follow called This Week in Virology. And one of it's basically 4 virologists talk every week, and they record it, and it's super. But they were talking about explaining some of the SARS-CoV data, and they said, there are 4 equally possible, non-mutually exclusive possibilities, and they were explaining a different phenomenon, but things that could be the reason. The Johnson & Johnson vaccine could have a different, the gene that they put in there could be a different part of that S spike protein. Okay, so the thing they are having your cells make could be slightly different, and your immune system could react differently to the two things. Thing number two is that, you know, you're delivering things differently. So you're delivering DNA in a viral coat in the Johnson & Johnson vaccine, and you're doing it with, you know, a virus that causes, you know, mild colds in people, and so that may not that may not stimulate the immune system as much as the combination of the lipid and the RNA that is the Moderna and Pfizer vaccines. So there's lots of little different possibilities. And again, we are comparing apples and oranges, right? We're comparing a 1-dose regimen with a 2-dose regimen. And what Johnson & Johnson found is they actually had better efficacy when they gave a second dose, but they chose not to for those sort of broader reasons, because when you're putting this much money in as a corporation, you really only have one shot at a time. So they had to decide, they had to make a decision as to what their approach was going to be. And I think it was pretty well known because, and the New York Times has had some good articles on this, that the Pfizer CEO was like throwing all his eggs into being the first, you know, into that basket. And so Johnson & Johnson may have decided, well, we're not going to be the first, but there's this gigantic, you know, billions and billions of people market that we could capture that we know Pfizer is not going to capture. And there's added benefit, I think, of doing moral and ethical good by developing something that is, you know, more widely available to everybody. So I don't know, Jill, do you have anything to add to that?
I think you just answered that question beautifully. So I'm going to just leave that at that. So I saw we have a few more questions in the chat, so I can take the next one. So the next question is, does the vaccine prevent you from spreading the virus to others? So I'll handle that question. That is a mixed bag question. So technically, you know, an individual who's vaccinated, that vaccine is not totally going to prevent that virus from replicating in your body. The only thing the vaccine does is makes your immune system respond faster. Because when we come into contact with an infectious agent like a virus, there is always this incubation period between the time we come into contact with the virus and when we start to have signs and symptoms that we're sick. So in unvaccinated individuals, that time, the time we start to show signs of being sick, that's when our innate immune system is responding. In a vaccinated individual, your immune system is going to start responding a lot sooner in that infection, but that doesn't mean that virus isn't replicating inside of your body. So while being vaccinated will certainly reduce your ability to spread the virus to others, it is still really important for people who are vaccinated to make sure that they are wearing their masks and practicing social distancing. Yeah. Because just because you're vaccinated and asymptomatic doesn't mean that when you cough in someone's face, that person won't be susceptible. So it's a bit of a dramatic answer, but long story short, it will help, but wear your mask.
Yeah. And the other little piece of information is that Pfizer just released information today that their vaccine prevents asymptomatic cases of COVID. So again, if you're one of those people who've had the Pfizer vaccine and you still manage to contract the disease, you'll know it. So you'll know to stay away from people. So we don't have to worry about this case where we have healthy people walking around breathing on people, but we still, again, we have to wear our masks because, as Jill said, there's this little period of time before you really notice your symptoms where you could be. There was a question I saw in there about the statistics. And so because the data was, everything was done so quickly. And so one thing, hopefully you may have noticed when I was talking about the vaccine development timeline, I used the phrase emergency use authorization. I did not use the phrase approved. Okay. So these vaccines, the 3 vaccines that we're administering to people all over the US right now are not approved vaccines. They've been given emergency use authorization. So in the case where somebody's statistics, isn't quite right, okay, the FDA is still looking at that data. And so, are they going, does anyone expect these things not to receive approval? At this point, so many millions of people have been vaccinated, and that it's highly unlikely that there's any statistical anomalies that would prevent that. However, the FDA is going to do its due diligence. And the other thing that I think has been a really cool and exciting development with the coronavirus vaccines is that because everything went so quickly, because we're trying to do things on such a massive scale, those of you who have been fortunate enough, like Jill and I, to have gotten doses of the vaccine, when you go, you are encouraged to register with this app where they check in on you every day, and then after a period of time every week, and they ask you, how are you feeling? What are your symptoms? So they're constantly studying how people respond to these vaccines, and anybody who gets a vaccine can be part of our understanding of how well they work.
So Kelly jumped over a question in the chat, and then I see we have a hand raised, so we'll get to that one next. So the question from the chat that I see is a question about, are there mRNA vaccines being developed against the variants? So that's a great question. So Pfizer has actually, and I'm sure Moderna and J&J are looking at this too. They've been looking at, is their vaccine effective against some of these variants? And so far, it seems that they are. The variants, while the variants might be acquiring nuclear or base changes in their genome that result in small changes in the spike protein, none of those changes have been major enough to affect the efficacy of the vaccine in any way, shape, or form. That being said, there's nothing to say that novel variants won't emerge that might be substantially different, in which case that would require a redesign of the vaccine. Fortunately for us, coronavirus is a relatively slow mutator. It seems to be acquiring mutations at around a rate of 2 mutations a month, which is slower than some, faster than others. And so I think it highlights really how important it is that we all do our part to limit spread right now. And that's the best thing that we can probably do. Limit the spread, get everybody vaccinated, and let's reduce the number of variants that are popping up. So let's go to the who has their hand up here?
My
Frederick
dad.
Ryan.
Hi, Dad.
This is Fred Ryan in North Haven, Connecticut. And my question is more of an ethical one rather than technical. Were any of these vaccines developed using cells from aborted babies?
So, Jill, you want to take that, or should I? Because that's it's been an issue that's been raised, a question that's been asked in the media. So.
Well, it depends. It's your dad asking the question.
Yes,
so
I can answer it. So the answer to your question, the way it was asked, is 100% no. All right. Now, there are what we use in the lab, okay, are cell lines so if we want to grow cells in a petri dish, okay, there are certain standardized cell lines, and there are only cells, after they grow and divide and a few times, will eventually die out unless they have some special features. So the cells that people use in laboratories for most common types of that preclinical research can be from the earliest and most commonly historically used cell lines are from cancer tumors that have acquired the ability to just keep dividing and growing forever, but also from embryonic tissues, okay, that also have this special feature that they can keep growing and dividing. So there are some companies that did some of their very, very early testing using a cell line that is it's an extremely common laboratory cell line, and the origin of that was an aborted baby that was in the 1970s, so there's actually a website where they have tabulated like company, vaccine project, whether they've used this, like what cell lines they've used, what animal models they've used, and so everyone can go and look. As far as I can tell from the Pfizer and the Moderna, I think each of those had maybe one very early experiment done with it, and from the scientific perspective, the vaccine itself, after the fact, they have enough, data to not use the data from those experiments in their application for approval. The other way, just personally, that I look at it, regardless of what your feeling is about abortion, rather than have that aborted fetus have been able to do nothing for the world, the fact that these cells were able to be used to save so many lives, from my perspective, is something worth honoring.
All right, so we're at 8:45. So I think we should work towards closing out. But actually, I have a question, which is a question about science communication. The issue of vaccine efficacy is been in the news a lot, and probably every day I see a journalist trying to explain this to the public, because when they see like the Johnson & Johnson 66%, they go, oh my God, I don't, what if I'm one of the 44% that's going to die if I take this vaccine? And you've explained it really, really well, right? Yeah. That actually you're not, right? And so there's kind of a discrepancy between the way the researchers describe effectiveness, right, and what's important to them, or at least what they're emphasizing, and what's important to the average person, which is not dying from COVID. Has there been, or do you think there will be, any rethinking of sort of what data is emphasized and commonly reported when talking about a vaccine? Because it would seem to me to be a very different thing if you said, get the Johnson & Johnson vaccine, 100% effective at preventing serious cases or deaths.
Well, so there's two things. Jill, you go first, and then if you don't cover one of them all. So
I have to say, for me, when I see a lot of people, researchers trying to communicate and express their results on the mass media, I see a lot of couching of the results. This may do that, or this might do that, or has the potential of. And I think it's a function of modern day mass media and just sort of the culture of distrust and sort of accusation that seems to be prevalent in our world today. I think that a lot of scientists or medical professionals are a little bit afraid of taking a hard stance on national television because you immediately sort of come open to attack. So for example, you can look at the data and you can say, this vaccine is 99.99% effective at preventing death. Go and get a vaccine. It's 100% effective. But then somebody comes up with this, strange case of this one person who was vaccinated, who, for whatever reason, they might have died. And like it, you just sort of open yourself up to those difficulties. And that's particularly prevalent with some of the scientists that are interviewed by mass media, because those scientists are either government officials or they are bigwigs in either a major research institute or a pharmaceutical company or some other sort of NGO. And so, those people, for someone like that to go on the record and say something and then be proven wrong after making such a strong stand, that has a real effect to erode the confidence that the public already has in scientists, which is already being eroded. Like you said, there's just a general distrust of science and scientists in general. I mean, how many times have people said to me, oh, the cure for cancer is out there, the pharmaceutical companies are hiding it? And no, it's not true. And scientists, we're not getting rich. We're not getting kickbacks from pharmaceutical companies to hide the results. If we did, I wouldn't be here, doing my job. So I think that's a big part of it. Kelly, what do you have to say about
this?
I
think the other little aspect is that Jill addressed the thing one that I was going to say, which was what scientists would say, and thing two is that the data is out there. It goes out in press releases, it goes out in filings to the FDA, and journalists, physicians even, who are not experts in infectious disease, but they have an MD and they can read the New England Journal of Medicine, they can look at the data. It doesn't mean that they understand it, that much better than your average person who, majored in biology in college. I think, Jill, as someone who's spent her, her whole professional career studying, studying microorganisms, can tell you that you learn a lot in graduate school and teaching things that medical students never ever learn. So one of the things that I find frustrating is that you have people who are making statements who do not have the expertise, but they are members of the medical community. So their word has a certain amount of weight. So, scientists can put their data out there, but there are media outlets that want clickbait headlines. There are, medical practitioners who either have their own agenda or who simply look at the data and it says something obvious to them, but they don't have the training to know that their interpretation is wrong. And so it's not always nefarious intent. It's quite often just not having the expertise. And those, again, because of social media, because of the internet, information can spread wildly, and it is, it's a really difficult job, not just in science news, but with any news, to really filter out and determine, whether you can trust the information. So.
Fantastic. Thank you so much, Kelly, Jill. Thank you so much for your time and for lending your expertise to us. It was super informative.
You can tell we love to talk about this stuff.
No, it's great. I mean, there's, it's such a great story, and it's such a, it's so important to sort of understand this, but it's also wondrous. Like it's just incredible. And that's why I asked about the effectiveness thing, because I think it's like coloring what is actually a remarkable achievement that we should all be celebrating. And it's just frustrating that, it's being undermined, but
it's a real question. I mean, it's actually a real question, because I was asked by a colleague today who has a sibling who lives in a state where you can choose to get Pfizer or Moderna, or not Pfizer or J&J.
Mm-hmm.
And they're having this sort of family debate about which one she should get, because, oh, well, I only want to do the one shot or whatever. Talking, with the person who asked me the question, I'm like, listen, have them do what they're going to do, because either way they're safe.
Yeah.
Right. Yeah. Is there a slight chance that she gets a bad cold, or is more likely to get a bad cold if she has the Johnson & Johnson one? Yes. But if she was already exposed to COVID in her environment, but she didn't get sick, her immune system may already be primed. And so the Johnson & Johnson may be like a second exposure to the virus for her. So it may be the exact same thing as getting Pfizer or Moderna. So it's not, it's the important thing is that, everyone takes advantage of getting a vaccine, no matter what it is, when they have the chance, and that everyone keeps on doing the social distancing measures, because every layer prevents the virus from being able to replicate and copy more times. And the fewer times we let it copy, the fewer mutants there will be and the fewer variants. So it really is super important.
And the ultimate moral of the story is that we're 6 months away from being out of this, thanks to all these vaccines. If we can just all buck up and just do it for another 6 months, we're going to save us another 2-3 years of heartache and pain. Mm-hmm. So just, I don't know, do what you got to do, grab a bag of cookies and just eat the cookies and just, Netflix, do what you have to do. Just get a vaccine as soon as you can get it, and just socially distance and wear your mask. And by a few months from now, things will be better.
Yeah. So.
Thank you.
All right.
Thank you so much. That was great, and a great note to end on.
Yeah, and thank you for everyone for staying with us so long. We had a big crew for 2 full hours of
really great.
We're all sick of by now, right? COVID.
But it was such a nice, thorough discussion of these questions that we're all thinking about right now, and you explained all that stuff just so clearly. So it was great. Thank you.
Good, excellent. Thanks for the opportunity, Doug and Sally.
Thanks for making the time, because I know you guys are so busy. So it really means I'm so grateful that you
not anymore. But
okay. All right.
Thanks so much. Good night, all. How you doing, Jill? You're muted. All right.